Hello Medicose, welcome to the channel. Today we are going to talk about excitation-contraction coupling across the heart muscle. It's a bit like an electrical storm, but instead of lightning we have calcium ions sparking the muscle fibers. It all starts with an electrical impulse, the action potential, which travels across the heart muscle like a wave. Imagine this impulse as a spark traveling along a fuse. This fuse is a network of specialized fibers called T-tubules, which penetrate deep into the heart muscle cells. These T-tubules are crucial because they carry the electrical signal to the inner depths of the muscle cells. As the action potential races along the T-tubules, it triggers a series of events that culminate in the contraction of the heart muscle. This process ensures that the heart beats in a coordinated and rhythmic manner, effectively pumping blood with every beat. Now let's zoom in on what happens when the action potential reaches a T-tubule. Embedded within the T-tubule's membrane are specialized proteins called L-type calcium channels. These channels act as gateways for calcium ions, much like doors that open and close. When the electrical signal arrives, it causes these L-type calcium channels to swing open. This opening allows a rush of calcium ions to flow from the outside of the cell to the inside, down their concentration gradient. This influx of calcium ions is crucial because it serves as the trigger for the next stage of the process, the release of even more calcium from the cell's internal calcium storehouse, the sarcoplasmic reticulum. We'll explore this in the next section. Inside the heart muscle cells, we find a specialized structure called the sarcoplasmic reticulum, a vast network of interconnected sacs. Think of the SR as a vast reservoir, storing a large amount of calcium ions. The SR's role is critical in muscle contraction. As calcium ions flood into the cell through the L-type calcium channels, they trigger a remarkable chain reaction. The incoming calcium ions bind to specialized proteins called ryanodine receptors located on the surface of the SR. These ryanodine receptors act like floodgates, controlling the release of calcium ions stored within the SR. This binding event causes the ryanodine receptors to open, releasing a flood of calcium ions from the SR into the cell's cytoplasm. This process is known as calcium-induced calcium release. The initial influx of calcium from the L-type channels triggers an even larger release of calcium from the SR, creating a cascade effect. Section 4. The Plateau phase, a balancing act. After the initial surge of calcium ions, the heart muscle cell enters a phase called the Plateau phase. During this phase, the electrical potential across the cell membrane remains relatively stable. This plateau is crucial because it prevents the heart muscle from relaxing too quickly, allowing for a sustained contraction. Maintaining this plateau requires a delicate balance. While calcium ions continue to enter the cell through the L-type calcium channels, other channels work to remove calcium from the cytoplasm. The most important of these are the sodium-calcium exchangers, which pump calcium ions out of the cell, and the Cerca pumps, which pump calcium back into the SR. This balancing act ensures a prolonged availability of calcium ions within the cytoplasm, sustaining the contraction of the heart muscle. The plateau phase highlights the intricate control mechanisms that govern the heart's rhythm and strength of contraction. Section 5. Calcium's dance with troponin. Calcium ions flood the cytoplasm from outside the cell and the SR. How do these ions cause heart muscle contraction? They interact with a protein complex called troponin. Troponin is a complex of 3 proteins on the thin filaments. These filaments are made of actin, crucial for muscle contraction. Imagine actin as a rope ladder with troponin as locks. Calcium binds to troponin C, causing a shape change. This change unlocks the actin ladder. Binding sites on actin blocked by troponin 1 are exposed. Section 6. Relaxation. Calcium retreats. As the action potential subsides, the influx of calcium ions through the L-type calcium channels slows down. The Cerca pumps on the SR membrane continue to pump calcium ions back into the SR, gradually reducing the calcium concentration in the cytoplasm. As the calcium concentration drops, calcium ions dissociate from troponin C. This dissociation causes the troponin complex to revert to its original conformation, blocking the myosin binding sites on the actin filaments. With the binding sites blocked, the myosin heads can no longer attach to the actin filaments, and the muscle relaxes. This relaxation phase is just as crucial as the contraction phase. It allows the heart chambers to refill with blood, preparing for the next heartbeat. The cycle of contraction and relaxation, driven by the ebb and flow of calcium ions, ensures the heart's continuous pumping action.